Hello my dear students, welcome back. In this session I am going to discuss about the structure of Embryosac. In my last session I completed megasporogenesis and development of female gametophyte. Now I am going to discuss the structure of Embryosac or Embryosac, which is also called as a female gametophyte. So this is a mature female gametophyte or mature Embryosac. So what are the different parts? So let me list down what are the different parts of mature Embryosac. There are three parts of mature Embryosac. The first part is called as egg apparatus. The first part is called as egg apparatus. So let us see one by one. So first egg apparatus, as I have shown in the diagram, this egg apparatus is found towards the micropylar end of the ovule. So egg apparatus consists of two types of cells. It has two types of cells. First type are called as synergids. These are the smaller cells of embryos, smaller cells of egg apparatus. These are the smaller cells of egg apparatus. As we can see there are two synergids present. Two synergids are present, and these synergids consist of a special finger-like structures, which are called as filiform apparatus. Special finger-like structures called as filiform apparatus. This filiform apparatus helps in absorption of nutrients. This embryo sac is embedded within the nucellus tissue. So therefore, this filiform apparatus will absorb nutrients from the nucellus tissue. That is one function. Another function, second function, this is the first function where these finger-like structures, filiform apparatus, help in absorption of nutrients. Now the second function is synergids also help in guiding the path of pollen tube. So pollen tube, when it enters into the embryo sac, it always enters through the micropylar and passing through the synergids. So they pass through the synergids and then they reach up to the center of embryo sac. So these synergids will help in guiding the path of the pollen tube. So there are two important functions. One is it is helping in absorption of nutrients, and the second one is it helps in guiding the path of the pollen tube. If we have a closer look at the synergid structure as in the diagram, we find there is a vacuole present towards chalazal end of the synergids, and filiform apparatus are present towards micropylar end. And egg cell also, if you compare egg cell and synergid, this nucleus is present towards the micropylar end, and vacuole, this is the vacuole of synergid, it is present towards the chalazal end. Now let us see about the second type of cell present in egg apparatus. The second type of cell present in egg apparatus, that is the actual female gamete, or it is called as the egg cell. Egg cell, or it is the female gamete, and it is the largest cell of egg apparatus. It is the largest cell of egg apparatus. As we can see in the diagram, its nucleus will be towards chalazal end. That means it also has a vacuole. That vacuole will be towards micropylar end. These are the structural differences between the synergids and the egg cell. Now what is the function of egg cell? Since it is a female gamete, it fuses with male gamete. And this process is called as a syngamy. That means it participates in syngamy, and that results in formation of zygote. So that is the egg cell. So egg apparatus has two parts, two cells. 2 synergids are present and 1 egg cell is present. Now let us see the second part of the embryo sac. This can be called as the second part, antipodals. Antipodals, as we have seen in the diagram, they are present towards the chelazal end. They are present towards chelazal end. They are present towards the chelazal end. And these are 3 in number. 3 antipodals are present, and these are smallest cells of embryo sac. They are smallest cells of embryo sac, and these are also called the vegetative cells of embryo sac. These are smallest cells and these are also vegetative cells of embryo sac, and they may or may not participate in fertilization. They do not participate, but they degenerate. They are vegetative cells, they are called as, because they do not participate in fertilization, and they degenerate either before or after fertilization. So what we can say, they degenerate either before or after fertilization. Either before or after fertilization. So this is about what are the antipodals. Now next let us see the third part of the embryo sac, that is called as third part of embryo sac, that is central cell. Central cell, it is the largest cell of embryo sac. Central cell is the largest cell of embryo sac. The entire portion excluding antipodals and egg apparatus, the remaining entire portion, that is called as a central cell, and it consists of 2 polar nuclei. It consists of 2 polar nuclei. How are these polar nuclei formed? We already discussed in the development. These 2 polar nuclei, they migrate from the 2 ends, micropylar and the chelazal end. So these polar nuclei are haploid in nature. They are haploid, and these 2 polar nuclei, they fuse just before fertilization, or just they fuse before the entry of pollen tube, before fertilization or entry of pollen tube. So just before entry of pollen tube, when they fuse and they form diploid secondary nucleus. So this diploid secondary nucleus will fuse with the male gamete, another male gamete. So what will this form? It will form a primary endosperm nucleus. And later this primary endosperm nucleus will give rise to endosperm. It will form endosperm. So this central cell, we can say it is the first cell of endosperm. And in the central cell, the ploidy, 3 types of ploidy we can find. So next point I am going to write here about central cell is the cell, it shows 3 types of ploidies. So what are 3 types of ploidy? First it is haploid, and then it becomes diploid, because secondary nucleus is formed, then it becomes triploid. And after fertilization, this secondary nucleus or the central cell, it becomes triploid. So 3 types of ploidies can be observed in this cell, and it is the largest cell of the embryo sac. So this is the structure of a mature embryo sac. Now This is also called, this embryo sac is also called as polygonum type of embryo sac, because it is for the first time observed in polygonum. It is also called monosporic embryo sac. Now, say in one question what we may expect here is regarding the size of these cells. We can give a descending or ascending sequence of these cells. So suppose we give the ascending sequence of these cells in terms of their size. Ascending sequence of size of these cells of embryo sac, of the cells of embryo sac. So this can be, you can get a question. So what is the ascending sequence? We start with the smallest cells. So what are the smallest cells? First the antipodals. After antipodals you will write next the synergids. After synergids it is the egg cell, and finally it is the central cell. So this is one type of question you can expect here. The previous previous video I already discussed about the divisions and which type of divisions and what type of meiotic, mitotic divisions can be asked. Okay, my dear viewers and others, that's all for now. In my next video I will be coming up with next topic, that is about the pollination. So in sequence I want to complete about this topic, and then one by one I am going to discuss even objective questions. So thank you very much for listening. Please do share and subscribe to the maximum extent. Thank you very much. Have a good day.